cell. Pluripotency is where many of the cells can come from a cell. So, yeah, totipotency, which animals don't have. We have pluripotent stem cells, but not totipotent stem cells in our adult body. Oh, no change, okay. In gene therapy, a defective gene can be replaced by a normal gene. If successful,
I think it's the one I put, but no.
very good.
Got it.
It was obvious. Genes on fire. What are some impacts of genetic engineering of plants? All of the above. They are all possibilities for changing phenotypes in genetically modified
I knew
plants.
Taste is one of them, but I just got the
Cell on top and on fire. What are two ways a gene can be delivered for gene therapy?
I think it's the virus. The virus is the most
Yes, and you're on. I think it's virus. I think you're right, actually.
Oh my gosh, yay! Oh,
Liposome and virus. Yep, so liposome is that fatty body that mimics a cell, that we can deliver the contents of that to a cell, animal cell, and we can also use viruses to deliver a gene into an animal cell. Lipoprotein is just a protein that's found in the cell membranes, and we don't use bacteria to deliver gene therapy, which is to people. Alright, IL still on top. Biolistics uses particles of
gold and titanium. Gold. Oh yeah.
So it's gold or tungsten. We don't use both together. We use one or the other.
I think it's titanium.
And we don't use titanium. Change. DNA or RNA vaccines work by? Great. So we're delivering those nucleic acids for transcription and then, or translation, to produce what is usually a pathogenic protein to induce an immune response. Very good. Okay, nice. EG is on top now. The bialistic method consists of excellent. Yes, we don't use microinjection of plants, and biolistics is using nanoparticles to deliver molecules that are shot or fired into an explant so it can get through the cell wall. Good. Right. Change. 10. What would be a drawback to using a DNA vaccine? I didn't actually mention this in class. I just thought, see how you go. It's actually that it might integrate into the host genome. So when we've got a cell that we're delivering a vaccine to, and we're going to use animal cells to do this, if we're delivering a gene that goes into the nucleus, it'll be transcribed and then translated. The mRNA goes into the cytoplasm for translation. If the gene, the DNA vaccine, is in the nucleus, there's always the risk that it could integrate into the genome, and so becoming a permanent feature of the genome. Yeah. Okay, males on top. How is a whole gene delivered into a genome using CRISPR-Cas9?
Yellow. That's what I
Great.
thought, but
So the method to deliver it is with a template and then using homology dependent repair. If we're using non-homologous end joining, then we're going to learn about this more next week, but you actually mutate the genes and you knock it out. So what I've talked about to date about delivering a whole gene has been using a template with homologous DNA repair. Right, the G on top, DLM is on fire. Plant transformation can be done by shooting agrobacterium into explants. True or false?
yes, it's true. Short guys. 50-50.
50-50. The answer is false. We don't shoot agrobacterium. We infect explants with agrobacterium. We shoot naked DNA attached to nanoparticles, gold or tungsten. Okay.
Yeah, good.
Change on the leaderboard. One is on fire. What type of genetic disorder is the best target for gene therapy? Didn't talk about this either, but I thought just extend your thinking.
I got it.
Great, nice. Yeah, so logically, we want to have a target that's relatively easy to overcome. A single gene mutation is always going to be better than something that is multifactorial, which is genes and environment working together. So gene therapy for cancer, it's being researched, but it's harder to achieve than, say, gene therapy for something like cystic fibrosis. We don't usually use gene therapy to target mitochondrial conditions at this stage because of the number of mitochondria in the cell, and any chromosomal condition is going to be like big chunks of DNA, so too big for gene therapy. Good going at that. Okay, a bit of a change. Vinci's still on fire. Okay, Agrobacterium tumefaciens.
Yellow? Oh.
Yep, so it is a soil organism that, but the bacterium itself doesn't go into the plant. It's actually the tDNA that goes into the plant. So when it does that, it forces the plant to make OPEs, carbon and nitrogen, for it to grow. Good. Okay, CKT's now on top. Expression of a transgene in only the anthers of canola, which is a dicot, requires Yep. The answers are all in the question. So we want it only in the anthers, that's the clue. If we said anywhere in the plant, it would be constitutive, but we said only the anthers, so it means anther-specific expression. And canola, the clue I gave you is that it's a dicot. And remember last week I said dicot promoters work better in dicots, monocot promoters work better in monocots. All right, here we go. Podium. Third position, we have DELN, who's that? Nice, well done. Second position, VG, who's that? question. CKT, who have we got? Yeah.